Because I said what was on my mind. I made a lot of enemies. I made a lot
Welcome, everyone, to Science from the Fringe. My name is Bryce Nickels. I'm a professor of genetics at Rutgers University, and today I'm once again joined by distinguished virologist Simon Wain-Hobson for the second episode of In Defense of Virology. Simon, how are you doing today?
Very well, thank you. And yourself? I enjoyed our first one. It was a great conversation. Let's see what we can drum up today.
We got a lot of very positive feedback on the first episode, but we did get a notable comment from your sister who mentioned that she thought my background left a bit to be desired.
Yeah, you see, my sister's an artist, so if I want to sort of just needle my sister, I would say the background is a perfect 1920s Mondrian with one or two lines and two colors and minimalist 1920 art. She would love it if she really knew that was a Mondrian behind you.
Well, that's true, Simon. I didn't want to tell people that I have a very valuable painting in my house.
No, of course not.
So the premiere episode of In Defense of Virology, we discussed, we defined some terms. We talked about what dangerous gain-of-function research was and why it's so risky. We talked about how there's not just a threat that if you make a dangerous virus in a lab that it will escape, but that the information itself was presenting potentially just as big a hazard. Can you talk about that some more?
Yes, that's probably something for you and me, it's obvious, but it may not be obvious to everyone. So we think of having these viruses in the lab. You get a lab leak or someone infects themselves in the lab, walks the virus out because they're infected. And could start off a chain of infection, an outbreak or an epidemic. That's the virological side. But in the modern world, there's the genetic information. So everyone knows that we have our genomes, DNA genomes. Humans are DNA genomes, bacteria, mouse, plants, you name it. It's DNA. This is the molecule. There's one little aberration to that, and in virology, we have viruses with just RNA, as opposed to DNA. So we have viruses like measles, like rabies, dear to Louis Pasteur, and flu. And then we have DNA viruses like papillomaviruses, herpes that gives you the lesions around your mouth, and many others. And so we have these two genomes, different types. And with the explosion of technology starting from the late 70s, now virology is determined by sequences, and you can understand the virus through these genetic sequences. This is long chain of building blocks like TCG, AC, TAG, GCC, etc., that can go on for several thousand building blocks. And that is the blueprint of a virus. With that information alone, just a sequence of 4 letters, T, C, G, or A, in just endless combinations, that information is enough for someone to resynthesize the genome of that virus. And once they've resynthesized the genome as a molecule of DNA, put it into, say, a bacteria, if it's a virus of bacteria, or put it into human cells and recover virus from that. So, for example, if I said, hey, Bryce, we've just identified a new virus and sequenced it, I could send you through internet the sequence, and you could reproduce that and make it in your lab. And all that passed between us was a PDF with that sequence attached to an email, and you could do it in your lab. So scientists do this all the time, but this is maybe not too well understood, because the scientific process also makes sure that this information gets published, and you have to show that you're a good scientist, you have to make sure this sequence is in a database. accessible, because this is the open access which has been so good to advancing science and helping collaborations between scientists. But that information could be taken by a lab in Russia, Iran, North Korea, and therefore this needs some thought, because we need to realize that this information we're putting out there free of charge, in fact, courtesy of the U.S. taxpayers, and the people listening to this are going to be mainly U.S. taxpayers. This is there, and I think we need much greater discussion as to, well, what should we put out there? Should we put out all this, or should we just put out some of it? And just one little thing is getting DNA into cells is not difficult. It was worked out 30, 40, 50 years ago. So this is absolutely trivial technology to do. In fact, it's rather fascinating. You take DNA, which has got a long molecule with lots of phosphate on the edge, and you add calcium. Well, your listeners know that around the sink and in the toilet, you get calcium phosphate deposits because it's insoluble. So when you add the calcium, this makes a sort of a mass, and the cells just gobble it up. So it's actually rather simple. And so it's trivially simple to get to DNA. And this was shown by a gent back in, I think the first time it was 1972. He showed that you could take the DNA from a thing called adenovirus that gives you respiratory and nasal infections, and he could get the virus into cells by adding calcium to it. So this is rather simple, terribly simple science. But so the information is going around the world, and there's not enough discussion. In fact, there's no discussion, in my humble opinion.
You mentioned that there are RNA viruses and DNA viruses. So for an RNA virus, can you explain how RNA viruses can be regenerated because you can purchase DNA sequences?
Yes. So you could synthesize the RNA if you want, but there's a much simpler way. You would synthesize the sequence is its DNA form. You clone it into a circular DNA molecule, which allows us to copy that DNA into RNA. So the cell would do it. And in fact, this is what cells do. Viruses are just parasites, as you know. So if you get, say, something like a herpes virus into a cell, the DNA gets into the cell, and then all the cell machinery starts copying that genome, copying it into RNA, and the RNA gets made protein. Or if it was an RNA virus like polio or influenza, that RNA would be immediately made into protein. So the cell does it for you. And so the same thing, you'd synthesize the RNA as a DNA form, and then get it copied into RNA, and the cell would do it for you. So all the hard lifting is done by the cell. Again, at the time, it took a lot of thinking and puzzling and experiment. Now it's trivial. You can do it for just about any virus you want.
I want to talk a little bit more about terminology. One of the terms of this space that we haven't discussed is something called DURC. Can you explain what DURC is?
Yes, it's not a very nice sounding word. DURC, it's a hard sound. Yeah, DURC, dual use research of concern. The obvious example is nuclear technology. So you can use nuclear technology to make nuclear energy. So particularly today, it's zero carbon emissions, and it can keep us going for quite a while. But of course, you can make atom bombs and then hydrogen bombs, and then put it, dirty bombs. Ultimately, put it onto missiles. So it has the same technology can give rise to good nuclear energy, clean nuclear energy, although you've got the disposal of the waste products, or you could go into the military side and you use it as an offensive weapon or as a deterrent. So research of this form is called DURC, and you can ask yourself, well, artificial intelligence, it could bring great boosts to the way we run companies, help people, and you can use ChatGPT and whatever, GPT, whatever. But it can be used, and knowing how the way the human mind works, you can be sure it will be used and be militarized in some context. So you can say, to what extent should we do this, or should we stop? Or there's a discussion going on in what's called mirror life. This is basically takes us back to DNA. DNA has a particular form, because the way the atoms are all organized, and you can make a mirror image of that chemically. And for years and years, this was a wonderful idea, very elegant. So you have a left hand, you can say, well, with a mirror, I've got a right hand. And so if this is the biological form, this one doesn't exist. But then you say, out of curiosity, well, wouldn't it be amusing to make this one? And 20 years ago, that was a fabulous dream and fascinating, and it piqued curiosity. But now we can start to do it. And so there's this discussion as to how far should this information and this technology be pushed. And there's a movement to get it stopped. It's not stopped, but there's a discussion, an open discussion going on, which is great. As you know, in dangerous gain of function, there hasn't been a good open discussion anywhere. But there is a discussion, and we will see how this goes and how the dust settles. But I think it's going to, some sort of curtailing of this research is necessary. So this is a DURC. So now it gets to our beef, which is the dangerous virology. So you can say, yes, well, the technology can make a new novel virus. This is dangerous. But what's the upside? Because pluses and minuses, people understand that. The good side, the bad side, the good news, the bad news. Where are we? And I have, in my essays, I tried to show that this dangerous gain of function research that was sort of crystallized in 2012, there are no upsides. There are no upsides, because they said this would help us predict the next pandemic. Remember, in 2012, the next pandemic was COVID. Not only it was a totally different virus, and now Dr. Kawaruka says this flu virus is not a threat for humans. We haven't got the foggiest idea what the next pandemic is going to be. We know it's going to happen, and it will probably happen in our lifetime still. Maybe not so much me, but certainly you. And it will be in the lifetime of your children, of your sons. So these are going to happen. Which one, we do not know. So I've argued that there are no upsides. So this work is not even DURC. It's not even dual. It's not even nuclear power, which can say, well, hell, these multiple warheads, I mean, nasty things, but at least we got carbon zero energy. We have no upsides for this dangerous gain of function. We just have the downsides. So it's not DURC, it's SURC, it's single-use research of concern. It's madness. There are no benefits. We're doing it because it can be done. It can be done. Well, wait a minute. There are two things. Firstly, we can clone humans. We could, but it's been banned. I think manipulation of human embryo cells, to choose, say, skin color or blue eyes or whatever you want or whatever, that's out. And I think this should be out. And by the way, to any doctors listening, when they got their medical doctorate, they had to say, first, do no harm, the Hippocratic Oath. So doctors are beholden not to do harm. It's in their sort of medical 101. But scientists, PhDs, don't have to take Hippocratic Oath. And they think they can basically do what they want. This is wrong. There's precedent for it. And this dangerous gain of function work is simply madness and should be stopped. And there are no benefits for humans. The DURC thing, in my opinion, in biology is not talked about enough. It's the bad news. No one wants to talk about bad news. Everyone wants to talk up the good news.
So the single use in your new term, single use research of concern, is that single use for bioweapons use? Is that what the downside only?
Well, it's the downside. So if you, I like to talk about making, it's just something very easy to understand. If you make the world a more dangerous place, then you should be asking yourself, what am I doing? So we've got enough pathogens on the planet to keep microbiologists for another 50, 100 years, okay? We haven't got a good vaccine for tuberculosis. Malaria is still causing tremendous numbers of deaths. Hepatitis C. And then we see, out of the blue, COVID, 20 million plus. And this is just going to come. It's part of what people have called the arms race between microbes and us humans or animals, because it's not, there are tons of viruses in animals. It's not just us. And this is ongoing, and this is part of the natural world, and it's always been this way. So we've got our work cut out, and I don't think we need to make new ones. And on top of that, if there was some benefits, we could then go into this discussion. Well, yes, maybe let's have an open discussion, Bryce. But as I've analyzed this, I've looked at it, and in my opinion, there's no upside to it. It's not DURC, it's SURC. It's even worse.
One of the problems in the COVID-19 discussion about where it came from has been the fact that scientists have tried to talk over the public. And so you said before that talking over the public doesn't work. It just creates suspicion. Can you talk about that a little bit more, and perhaps talk about how you have some experience in this area?
Yes, well, I've sort of cut my teeth on, virologically speaking, in HIV going back to the mid-'80s. And so I grew up fast because it was hardball. And the HIV people got it all wrong in the beginning. They came in with their elbows wide and saying, we're going to do this. The Secretary for Health back at the time said we'd have a vaccine in 2 years. 40 years later, we don't have a vaccine. And there was too much hubris, too much noise. And the HIV crowd realized that the researchers were getting a very bad press. And it was through working with the patients that things suddenly started to change. And this was a great transformation. And be able to sort of have HIV people in those big science conferences and discussing with them. They became the best collaborators of the HIV community. The clinicians dealing with the patients and bridging between the scientist and the patient. And this helped HIV and the research towards getting the drugs and now the triple therapy. So the HIV people made lots of mistakes. But I'll give you one very clear example. There was a journalist who came out with a book called The River, and he made the link between the fact that the epicenter of the pandemic is the East Africa, the Rift Valley, and that is where the study of oral polio vaccination occurred back in the late 50s. And he put two and two together. He basically said, I think the origins of HIV has something to do with this vaccination campaign. So he wrote this long book, huge book. I think it's like 500-600 pages. And I was asked to review it by a journal. So I said to myself, sure, I'm an HIV. I'll find the weak spot. And I read the paper. I read the book. It took me 3 days nonstop. And I couldn't find the smoking gun. I couldn't find the crucial error. And so I wrote a book review, as you do. And I basically sort of said, well, it's actually not clear what's going on? So instead of trashing the theory, I got trashed because I didn't demolish this book out of hand. Okay? Some of the most vitriolic emails I've ever had came from virologists, a professor of virology who I will not name, in the US, by the way. I mean, if you that email, I mean, if I put it into, you remember Mission Impossible and put in your briefcase would go up in smoke because there was so much acid. So whatever. And we ended up organizing a meeting at the Royal Society. The daggers were drawn, the protagonists, and it was a slogging match of rhetoric and bad-mouthing. And so we got the meeting at the Royal Society in London in 2001. And we got the two protagonists there, the elderly gentleman who performed this trial, Professor Koprovsky, who was still alive and remarkably sharp at well over 80, remarkably sharp brain. And we had the journalists there, and we had people from both sides. We had scientists who had gathered more data, and the more science was coming in, and the meeting was totally open. So it was open to anyone and everyone, journalists, and at the end of it, we had a press conference. There were 5 TV crews there. The people were there, the journalist and Dr. Koprovsky under the cameras, and people were allowed to ask their questions publicly. What happened is that by that time there was much more data coming in, and we had tested, my lab had tested some of those very, very early vaccine lots and lots more virology, and it was clear that the virology didn't add up, and that the HIV had been around for much longer than we had thought. And it wasn't 1959, as some people suggested, or the late 50s. And the journalist didn't do a good job in public with and it was clear he didn't marshal his arguments terribly well. But the most revealing remark from me was a journalist from one of the TV crews. He said, well, it's pretty obvious to me, Simon. He said, thank you for organizing this with your colleagues, but it's pretty to me. If you had something to say, you would never have organized this meeting. That, for him, that was the most convincing piece of data. We would never have exposed ourselves. We would have covered up if there had been something to cover up. And he said, you played open house, and that was it. And at that meeting, there was a former UK government scientist who handled the mad cow disease. I don't know if you remember, Bryce, but and your readers, but there was something called mad cow disease that gave this from cows, that gave a bovine spongiform disease, and it was a few cases in humans called virulent Creutzfeldt-Jakob disease. And this was harrowing, and no one knew what was going on because we had never seen this before in humans. And he took, as the government advisor, they took the haughty top-down, leave it to us, we got the experts, we know what we're doing. And, of course, they mishandled it. And it was this meeting on the oral polio vaccines was 8 years later. And he said to me privately, he said, Simon, this is what we should have done. We should have had a meeting at the Royal Society and just told people, this is what we know, this is what we don't know. You think this, we think that. Can we discuss? Open. And we would have spared ourselves a lot of heat and angst, and we handled it badly. And this gentleman was a consummate insider member from the Royal Society, more highly qualified with, every box was check, and a brilliant scientist at the same time, excellent scientist. And he said that. So when, virology doesn't have to open up for everything and get into, I'm sure your readers would be bored with some of the details, but when it's like, oral polio, HIV origins, COVID origins, dangerous gain of function, this naughty virus seems to be breaking out somewhere. What do you think? The times when virology has to open up, And we just have to open up and say what we know and share actually some of the angst, because if this sort of stirs up your listeners, we are not untouched. We are I'm not sure news, but sometimes we're shit scared. I mean, with HIV, we used to call it, the scientists between us, we used to call it the Dark Ages, from 1983 up to the arrival of this triple therapy. The science was making wonderful gains, and the disease, the information coming, got blacker and blacker and blacker every year. And we were sort of saying, what's going to happen? We were, sorry to use the bad word, we were shit scared. And this happens. And so we have to share. And if we have our doubts, then you can be sure the public. And so there are times when we just have to open up. And the funny thing is, scientists can't do this. By and large, they can't do this. They hunker down like an oyster or a snail. They go back into the shell, and they keep everyone at bay. No. It's the bad news, but if scientists can't handle bad news, then who can handle bad news?
Can you talk about what Fauci's role was in that conference back in 2001? Or
No, he wasn't involved in that conference
Okay.
at all. The journalist was English, so the focus of attention was mainly in. But the scientists didn't like it at all, and they shot from the hip. And there was a gentleman called Peter Duisberg. He's a professor of virology in San Francisco at Berkeley. And early on, he said, well, HIV doesn't cause AIDS. And people sort of poo-pooed and said, Peter, you don't know what you're talking about. Lots of ad hominem comments. But people weren't bothered because Peter wasn't in the field. But the problem is that Peter got the ear of the president of South Africa at the time, President Mbeki. And Mbeki, therefore, because he had doubts as to the origins of HIV, said, well, why should I spend millions of South African rands helping or trying to treat AIDS when HIV is not the origins for it? And so that was the bad. So once again, the scientists had to get together, and I was involved in that. I was part of something called the Durban Declaration, because Durban from the city in South Africa. And we got this argued, I think a 20-point, giving all the data saying why HIV caused AIDS, what was the solid pieces of data, and in those 20 pieces, there wasn't a single piece of flaky information. The document was written with probably about 40, 50 people from all over the world, clinicians, immunologists, virologists. And then we got it published in Nature, and we got 5,000 people signing it. HIV doesn't cause AIDS. And that put the end to it. You see, when the virologists face up to the music, or when scientists face up to the music, and they explain why they think X, Y, Z, it's much easier for people to understand. Unfortunately, the top-down approach is still used, and it's fatal, and it gives rise to doubt, and it reduces the trust the public has in scientists, of which I'm one, and you too. And we need science because there are lots of diseases out there. And when the next pandemic comes, people aren't going to go for general thoughts about this and this. They're going to sort of say, okay, is there a drug for it? No, there isn't. Then what we have to do, get it. Do you have diagnostics? No, you don't. You need it. What are you going to do? Vaccines. They're going to say 1, 2, 3, 4, 5. They want the data. They want to be protected. They want to be protected, and they're not going to accept wishy-washy hand-waving arguments.
I will invite people to, I do think this HIV origin question has just emerged. And in the wake of COVID, there's a lot of people that are very skeptical of whether or not HIV had a research.
But
that's, I think, unfortunate. Well, I guess what I mean, Simon, is I'm just going to invite people to leave comments and questions they have, and we can maybe go over them
No,
later.
no problem. But if people want to comment that's their right. With pleasure, we're not afraid of comment. But this is, there's a pretty good consensus among virology and the data is very good. So if you don't like it, fine, but come up with very good arguments, please. Not wishy-washy, and not because the arguments are. It's really very strong. But yeah, it took time. It took us time.
Is that conference recorded? Is there a recording of that conference from 2001, or was there a summary written that anyone?
There was, but I think it was taken down after it was up for at least 6 years. But
Okay,
I
be
don't
useful to get
think
that again.
it was is there. So and practice is that put it that the virologists and the scientists are getting their they're cleaning up their act. There are conferences devoted to biosafety. You have all these biosafety officials trying to do the best they can, but there was a time when things were not regulated, as we all know. People put chemicals down the drain, and I'll give you an example. It's not going to.
What's that? It's not gonna.
No,
Some close calls. You want to talk about some close calls?
This is goes back to polio. So the oral polio. This is not the sulk, which was in an inactivated virus. This was live attenuated virus, and basically, this was the one that basically decimated polio across the globe and basically reduced oral polio vaccine to very, very low levels. And now it's a sort of a sniper job picking off the last few cases. But the oral polio attenuated vaccine made by Sabin, that you and I probably had a sugar lump. Certainly I did when I was young. So it was, you grew the virus, the polio virus, in fresh kidneys. So at the time, they used the kidneys from rhesus monkeys. Rhesus monkeys came from India. And in so doing, virologists started looking. They said as well, what if these animals have viruses? And they found a number of viruses in that. And in the literature, you can find the numbers like SV5 or SV40. These were found during people trying to find out, well, what if these monkey kidneys had viruses in? And for some reason back in the 60s, and I can't remember the details, my apologies to your listeners, there was a Brisbane between, there was a snaffles between the US and India would no longer allow rhesus monkeys to be shipped to the US. So looking for a source, in the US, they found African green monkeys. Which live in the savanna of Africa. And therefore, they started making this. This would be, again, in the 60s and 70s. Of course, HIV was discovered in 85. In 83, and published. In 85, the first HIV-like monkey virus was described in captive rhesus monkeys. And pretty quickly, they found a simian HIV distant cousin in African green monkeys. So we were using oral polio vaccine that probably almost certainly was contaminated by these African green monkey viruses, which are distant cousins to HIV. We didn't know at the time. We didn't know of even HIV back then, because we were learning virology and we're discovering slowly. It takes time, and we didn't have the means and the technology. Now we do, and so it was a close shave. But we know now that those green monkey viruses do not infect humans. We've tried and tried and tried to put it onto human cells. It hasn't been done. It's never worked, not even once. So it was a close shave. It was a close shave. That, of course, is not possible anymore. Absolutely out of the question. Can't be done. But so that was a close shave linked with polio vaccine and HIV. And so the act is totally clean now. You're not going to get an animal virus getting into a product. There's no way. No. But back then, the story was different. So yeah, close shave. These happen.
Well, we'll probably talk about a lot of other close shaves in future
Yes.
episodes.
And I think, and I would say, coming back in, some virologists aren't going to like me having said what I said then about that, but I'm not trying to.
Well, but who
And
cares, Simon?
this is
Who
public.
cares? You're not
But
making friends here. We're
no,
not making friends.
but if we're not honest with the public, if we're not, then they're going to go for it. Obvious. I mean, they're going to say, "Well, what are you covering up, and why?" And this is public knowledge. It's public knowledge. So when it comes to COVID origins and this gain of function, I mean, you realize this SURC sort of single-use research was supported by the NIH, and it could own, and which side was supported? It was the downside for erroneous logic. Now that's a bad mistake. Okay, nobody's perfect, and we're not going to throw the first stone. But that was, I think, a seriously bad. And some of us were asking for more open discussion, and we got pushback. It's not complicated when things go. Next virus hits, we just have to, and the public want to know, because they don't want to know all the detailed papers that come out in the Journal of Virology and Virology in a language they wouldn't understand, because it's so very technical and esoteric. It's typical of any specialist language. But when they want to know the big brushstrokes, and when something comes, we just have to give them the brushstrokes. And if we say, well, actually, we don't understand that, or this is a hole, we don't have it. As I said last week, we don't have any drugs for papillomaviruses. We don't have any vaccines. Hepatitis C, we have drugs. We don't have vaccines. And there are lots of things we would love to have to treat and get these things done. And we don't know when the next pandemic is going to hit. We do not. And if you deny that, if you say, that's too defeatist. Well, it's just looking at the data. I would love to be positive and reassure people, but it's just the way the chips fall. Maybe in 20, 30, 40 years, terrific. Right now, they haven't fallen that way. So we just have to sort of say things as they are. And it's when you don't say things as they are that actually this is actually even more dangerous. More dangerous.
So to sum up, and I'm going to put a pin, I'm going to end it here, our second episode, Simon, maybe, is it fair to say, if virologists truly care about protecting humanity and they want to get money from the public to do their research, they have to share knowledge openly, sort of maybe grow a bit of a spine, and perhaps most importantly, exhibit some humility and be open to public criticism. And, that will, at least based on what you described happened in 2001, that sort of ended a discussion, although it got brought up again because of the bad choices made in the wake of the COVID pandemic. So I guess with that, I'm going to thank Simon for joining me today for the second episode of In Defense of Virology. And I would like, if people have any questions or comments about anything we talked about today, please feel free to leave them. And maybe actually maybe we will take some questions. I don't know. We don't know what we're doing on future episodes, but I can see that you're gonna cause a lot of people to have some comments, Simon. If you're going to be talking about these old things, man.
What you're going to do? What you're going to do? What we want to do is want to help people understand, and so we will adapt. And if there are some questions out there that we can help answers with, etc. As I say, for HIV origins, you need very good data, but we're ready. We're there to help understand, because we have to share. This is our job. We're talking about virology with my, but if it was your area of genetics, I would help you to help people to get the message across, and we would try and find ways the information is not the format that you and I set up, is we're there to help people understand. And if that can help them reassure and see that, what we're doing is useful and help protect society and people from suffering, because, maybe in the US and France where I am, but, there's a lot of suffering from disease elsewhere in the world. Horrible. So we've got more than our work.
We
cut
do,
out
but
for
we
us. So
but we
we're
also need subscribers, Simon. We need people to share, like, subscribe, leave comments, because we're going to become major influencers in the science podcast space.
Well,
Just
yeah,
throwing
I know you. It's like, I'm probably old-fashioned. I don't like the word influence, but we're there to help. We're not there to sell, to convince, to sort of come off and make money out of people. Our service, or what our goal is, to help people understand. That's number one. Everything else is secondary.
And so with that, I'd like to thank Simon again. This has been Bryce Nickels on Science from the Fringe's In Defense of Virology. Take care, everyone.
Take care, and thank you for showing us that wonderful Mondrian painting once again, Bryce.
That's right. I'll be back in front of the Mondrian next time.
Okay, see you soon.
Bye, everyone.
Take care, bye-bye.